Hi everyone! Here is a discussion about replication, transcription, and translation process. First, let's recall the nitrogenous base pair rule. The DNA is located in the nucleus of the cell. If the cell dies or gets damaged, the cell undergoes cell division. The DNA makes a copy of itself in the process called replication in preparation for cell division. DNA contains the bases adenine, thymine, cytosine, and guanine. During replication, adenine pairs up with thymine. Thymine pairs up with adenine, cytosine pairs with guanine, and guanine pairs with cytosine. That is the rule in pairing nitrogenous bases during DNA replication. When the cell is healthy, one of the daily activities of the cell is to make proteins. Proteins are converted into enzymes, antibodies, hormones, and structural molecules. Our hair, skin, and nails are made up of proteins. The process wherein our cells make proteins is called protein synthesis. There are two stages of protein synthesis. The first one is called transcription. The DNA is copied by the RNA in the process called transcription. DNA is transcribed or copied by mRNA. The DNA has the bases adenine, thymine, cytosine, and guanine. The RNA doesn't have thymine. Instead of thymine, it has uracil. Adenine in DNA pairs with uracil in RNA. Thymine pairs with adenine in RNA. Cytosine in DNA pairs with guanine in RNA. And guanine pairs with cytosine in RNA. The only difference is that mRNA doesn't have thymine to pair with adenine. Instead, it uses uracil to pair with adenine. That is how RNA transcribe the DNA in the process called transcription. The second stage of protein synthesis is translation. The messenger RNA will enter the ribosome in the cytoplasm. The bases in messenger RNA will be translated by threes. Three bases are called codon. The tRNA, which carries anticodon, will pair up to the messenger RNA codon. Uracil in messenger RNA pairs up with adenine in tRNA. Adenine pairs with uracil, guanine to cytosine, and cytosine to guanine. There are three stages of translation. The first one is initiation, the second is elongation, and the third is termination. Please read your notes on protein synthesis for more details. Now let's practice. Say we have a hypothetical DNA template, and the DNA needs to replicate in preparation for cell division. Take note of the base pair rule. What will pair with thymine? It's adenine. What will pair with cytosine? It's guanine, right? What will pair with guanine? It's cytosine. This time, take a sheet of paper and try on your own. Write down the complementary base pair of the given DNA template. Please pause the video and try answering. Are you done? Let's check your answers. Do we have the same answers? Well done! You have performed DNA replication successfully. This time, the cell needs to perform protein synthesis. So, the messenger RNA needs to copy or transcribe a section of the DNA template. Given with that hypothetical DNA template, transcribe for the mRNA sequence. Get a piece of paper and give it a try. Please pause the video. In transcribing the DNA into mRNA sequence, please take note of the base pairing. Let's check your answers. Do we have the same answers? Great job! Now let's try to pair the codons in the mRNA sequence into its corresponding anticodons in tRNA. Take note of the pairing of the codons in the mRNA to its corresponding anticodons in tRNA. Now let's start. Get a piece of paper and try pairing the codons to its corresponding anticodons. Pause the video for your turn. Let's check your answers. Do we have the same answers? Great job! This time, let's translate the mRNA codons into a specific amino acid. In the protein synthesis module, you will discover that in the ribosome, you will find three sites. We have the acceptor site, P site, and the exit site. As the messenger RNA moves along the ribosome, transfer RNA moves to a specific site. One codon is read at a time. Translation begins when the codon is AUG. It signals the initiation or the beginning of the translation process. Therefore, the AGC codon in the mRNA sequence will not start the translation process. When the AUG code enters the P site of the ribosome, that is the only time when the amino acids start to build up. Amino acid is the building block of protein. Now let's translate the mRNA codon into amino acids. How do we do that? We get our genetic code table. The codon is made up of 3 nitrogen bases. In this case, the first letter is A, followed by U, and the third letter is G. AUG is a start codon, so it is the codon that will be first translated into amino acid. How do we find its corresponding amino acid? We look at this side of the genetic code, which indicates the first letter, and then the second letter is in this row, and the third letter is on the other side. Next, A can be found in this part. So that is UCAG. So, followed by the U part and G. So, we get the word methionine. Let's have GAG. So, the first letter is G. So, this part is G. And then the second letter is A. We look at on top of it, and the part where the two letters meet is where we can find its corresponding amino acid. So this time, it is glutamic acid. This time, let's try CAU. So C, the first letter can be found here, and the second letter is found beside. The box where the two letters meet is where you can find its corresponding amino acid. Again, the part where the first and the second letter meet is where you can find its corresponding amino acid. So this time, it is histidine. Please remember that we are using the mRNA codon to translate it into amino acid. It is not the anticodon. Try translating the rest of the mRNA codon into amino acid. Please pause the video and give it a try. Are you done answering? Let's check your answers. Once we reach an mRNA codon that is a stop codon, it signals for the termination of the polypeptide sequence. The polypeptide will now be released into the cytoplasm and be converted into a functional protein. I hope you learned something new. Thank you for watching.